Hello students, welcome to EPG Pathshala. I am Dr. Zinat Iqbal from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today I am going to talk about the module Mucoadhesive Drug Delivery System. We'll be highlighting the types of the MDDS. This is the part 3 and the conclusion of the series, and this is from the paper Novel Drug Delivery System 1. My dear students, in the last few of the modules, we have been trying to understand the concept of mucoadhesion. It is an important concept wherein we are talking about a mucosal surface, wherein we are attaching a drug delivery system. One surface is the biological surface, the other is an inanimate object, and when these two come together, the concept or the purpose of mucoadhesion is done. The mucoadhesive drug delivery systems can be targeted to different routes. Like, we can have a mucoadhesive drug delivery systems for the buccal patch. We can have a mucoadhesive drug delivery approach using the rectal route. We can have something for the vaginal route. Similarly, one of the major routes of administration is the oral route. Herein, we have a very simple system using the mucoadhesive polymers, which is targeted to be used for the adhesion on the mucosal membrane of the GI tract. The name is mucoadhesive gastroretentive drug delivery systems. In today's module, we will focus on the learning objectives. We will try to introduce what are gastroretentive drug delivery systems, primarily using the approach of the mucoadhesion, because the stomach would be the primary site of the action. Therefore, we would also like to have an overview of the stomach. We would also look into the advantages and disadvantages of such drug delivery systems. Then we will talk of the approaches of the GRDDS. Finally, we will have a major focus on the mucoadhesion for the purpose of the designing of the gastroretentive drug delivery systems. Later on, towards the end of the module, we will also try to highlight the various researches just by giving a few examples how they can be used to understand the mucoadhesion as an approach for GRDDS. Oral administration is the most convenient and preferred means of any drug delivery, and this is the most exploited, the most suited, and the most patient compliant route. Take advantage of this premise of oral delivery to be one of the most popular, and then amalgamating it with the usage of the mucoadhesive drug delivery domain, we generate something which is referred to as gastrointestinal mucoadhesive drug delivery systems. This system is highly suited for the drugs which have got short half-lives and show rapid absorption from the gastrointestinal tract and are eliminated quickly from the systemic circulation. Therefore, the need comes that these systems should be able to sustain the oral drug delivery, and therefore there is a requirement of release of the drugs slowly into the GIT tract. In such a scenario, the gastroretentive drug delivery system comes into the rescue, and this is supposedly a site-specific drug delivery for the delivery of such drugs at the stomach site. It helps in retaining the delivery system in the stomach, thereby releasing the drug in a controlled manner using the premise of mucoadhesion, and probably can also be modified for releasing the drug at the different absorption sites in the GIT itself. Stop. Overview of the stomach. It is well understood that until and unless we know the nuances about the site of delivery of the drug, we will not be able to understand the behavior of the drug delivery system. Before we get into the details of the gastroretentive mucoadhesive drug delivery systems, we would like to take a very quick overview of the structure of the stomach. Beginning with its location, it is located in the upper left-hand portion of the abdomen, just below the diaphragm, It occupies a portion of the epigastric and left hypochondriac region. The functions primarily, the main function of the stomach is to store the food temporarily, grind it, and then release it slowly into the duodenum. The stomach structure has got four main regions, primarily shown on the in front of you, consisting of something like cardia, fundus, body, and pylorus. Understanding the migrating myeloelectric cycle of the stomach. During the fasting state, an interdigestive series of electrical events take place which cycle through both stomach and intestine every 2 to 3 hours. Interdigestive myeloelectric cycle or migrating myeloelectric cycle is divided into the following four phases. The phase 1 primarily is a quiescent period lasting from 30 to 60 minutes with no contractions. This can be an equivalent of the resting phase. The phase 2 consists of intermittent contractions that gradually increase in intensity as the phase progresses, and it lasts about 20 to 40 minutes. Gastric discharge of fluid and very small particles begins later in this phase. The phase 3 is a short period of intense distal and proximal gastric contractions, 4 to 5 contractions per minute, lasting about 10 to 20 minutes. These contractions, also known as housekeeper wave, sweep the gastric contents down the small intestine. Phase 4. This is the short transitory period of about 0 to 5 minutes, and the contractions dissipate between the last part. The drug candidates for the formulation of gastroretentive drug delivery systems. As in other cases, there will be a suitability check for the formulation of gastroretentive drug delivery system. The drugs primarily can either be used for the local action, or they can also be exploited or explored for the systemic action, depending upon the intentions of the formulators. For local action in the stomach, we can have formulations which consists of antacids and drugs which are targeting the H. pylori infections. Similarly, we can have a particular drug which will only be absorbed in the stomach and exemplified by candidates like amoxicillin. Similarly, we can have drugs like furosemide, diazepam, verapamil, which are very poorly soluble at the alkaline pH, therefore need to be formulated into the gastroretentive drug delivery system focused at a particular portion of the GIT. The drugs which have narrow absorption window are exemplified by cyclosporin, levodopa, and methotrexate can also be exploited for designing into this system. The drugs which absorbed rapidly from the GI tract like metronidazole and tetracycline can also be used. The drugs which primarily degrade at the colon pH can deliberately be made to be formulated in a manner that they deliver the drug ahead of the reaching of the colon, and examples can be ranitidine and metformin. The drugs that disturb the normal colonic microbes like antibiotics against the Helicobacter pylori are very suitable to be formulated into a gastroretentive drug delivery system. We have just understood that the gastroretentive drug delivery systems can be formulated by using the drug candidates which has got typical advantages or typical properties. The gastroretentive drug delivery systems per se offer as a dosage form certain very important and peculiar advantages. These are: number one, they will guarantee an enhanced bioavailability. They will also offer a sustained drug delivery, and of course, they will additionally give the reduced frequency of the dosing. They are very suitable whenever we are requiring targeted therapy for the local ailments in the upper part of the GI tract. It will also reduce the fluctuations of the drug concentration. It definitely improves the selectivity in the receptor activation. It reduces the counteractivity of the body, and it also results in extended effective concentration. Finally, it can also offer an advantage where it minimizes the adverse activity at the colon part of GIT. Whenever we talk of advantages, there has to be the flip side to it. So we have also a series of disadvantages associated with this particular system. The first disadvantage is the drug substances that are unstable in the acidic environment of the stomach are not suitable. Systems requiring a high level of fluid in the stomach for drug delivery to float and work efficiently. It is not suitable for drugs that have solubility or stability problem in GIT. I think this point is very well taken because the site specificity is an important factor. We have to talk about the GI tract. So if there is a drug which is not suitable, which will have solubility or stability issue in the GI tract, so that becomes a very important disadvantages. So the drugs again which are irritant to the gastric mucosa are definitely not suitable. After understanding the various advantages and disadvantages and also having a fair idea about the suitability of the drugs, let us now discuss the various approaches which are available for formulation design in case of the gastroretentive drug delivery systems. We can have different approaches like that of using a high density material which will keep the drug system sedimented. We can also have a system where we can use polymers which are light in weight and which continue to remain floating on the GI contents. Similarly, we can use a system having the capacity of swelling and can use the polymers which are swellable in nature. The system so designed will swell and expand and continue to release the drug over the proposed period of time. The last one and the one which is of interest to us is the mucoadhesive and the bioadhesive systems. These primarily will rely on the capacity of the polymers to just probably have an intimate contact in the form of the dosage form with the stomach wall, remain adhered and continue to deliver the drug at the precisely controlled manner. Mucoadhesion as an approach for gastroretentive drug delivery system. I just mentioned that the behavior of the polymer and its capacity to attach to the stomach wall or the mucosal membrane will probably decide on its retention time. The mucoadhesive polymers are used in a manner either singularly or primarily in combinations to deliver a drug delivery system which has got a huge amount of mucoadhesion capacity and also which will be able to retain the dosage form at that site of action for prolonged duration of time. The adherence of such systems can be based on different mechanisms, and those mechanisms can either be based on the electronic theory, the fracture theory, or any other such theory which has been proposed depending upon the type of the polymer chosen. The mucoadhesive or the bioadhesive polymers which can be exploited for such purpose include the acrylic acids, including carbopol and polycarbophil. We can also use polymers like chitosan, which is a natural polymer obtained from the chitin source. Then we can have the gantrez polymer, which is the proprietary name for polymethyl vinyl ether or maleic anhydride copolymers. We can also exploit cholestyramine, tragacanth and sodium alginate for the purpose of designing the gastroretentive drug delivery systems. Dear students, just a while ago we had tried to understand that what are the various types of mechanisms which are present in the stomach or the GI tract. Because the stomach is a very dynamic environment, therefore it is a big huge challenge for the drug delivery system to remain adhered. There are certain limitations which has to be understood for any gastrointestinal mucoadhesive drug delivery system. The very first part is that because of the sheer presence of the acidic environment and sometimes because of the high turnover of the mucus, the bioadhesion is not a very reliable phenomena. So, the conclusion is that if at all the dynamic system is not allowing the adherence, then we will not get the therapeutic outcome which we are wanting to get. Then there is another limitation that it is not suitable for drugs that may cause the gastric lesions. Examples, if we have drugs which are the non-steroidal anti-inflammatory in nature, if at all they are given in the form of a bioadhesive or mucoadhesive gastroretentive system, what will happen? They will go and they will adhere to the inside of the stomach lining or the inside of the mucosal membrane. The outcome is because of the sheer problem of their irritation, the gastroretentive dosage form will not be able to stay there and therefore might be a complete and total failure. The drugs which are unsuitable for delivery in the strong acidic environment are again having a limitation, therefore they cannot be formulated into the gastroretentive mucoadhesive drug delivery systems. Then if you move on, as I've just said, that the stomach is a very dynamic environment. Therefore, the mucus secretion from the walls of the stomach will probably be never constant and therefore it can result in unpredictable adherence. Again, a failure of the dosage form. The bioadhesion systems in patients with typical problems of achlorhydria is not suitable as faster swelling properties are required and a complete swelling of the system should be achieved well before the gastric emptying time. This could be a limitation in a patient who suffers from achlorhydria. We will now discuss the various factors controlling the gastric retention of the dosage form. Gastric retention per se is the primary attribute based on which we can talk about the success of the dosage forms. The factors are summarized below. Beginning from the first factor is particle size. The particle size should be in the range of 1 to 2 mm to pass through the pyloric valves into the small intestine. The density of the dosage form should be in the range of 1 grams per centimeter cube to 2.5 grams per centimeter cube. The size should be greater than 7.5 mm in diameter. The shape of the dosage form could be either ring and tetrahedron devices with flexural modulus, which probably are the ones which are most suited for the gastric retention. Similarly, we can either have the single unit systems or we can have the multiple unit systems. The multiple unit systems are more preferable because of the predictable release profile and the co-administration of different units can also be encountered, and finally they have got larger safety margins. The food intake is again an important factor. As understood, the gastro-retention time will be much longer in case of the fed states. The nature and the calorie content of the food will also be an important factor. Indigestible polymers, fatty acid salts, increased calorie content leads to increase in acidity, which increases the gastro-retention time. Fat and protein meal also increase the gastro-retention time. Furthermore, the next factor is the frequency of intake. The GRT or the gastro-retention time increases 400 times due to the low frequency of MMC. The next factor which will influence the GRT or the performance of the gastroretentive drug delivery system is the posture of the patient or the consumer. It varies between the spine and the upright and the ambulatory state. Of course, while lying supine or probably not having any ambulation or movements will result in much prolonged gastroretention time. The next factor which has been highlighted is that of being of a particular gender. It has been reported that the females have got much shorter gastroretention time as compared to the male counterparts. The next factor is that of a particular age. The age beyond 70 years shows longer gastroretention time and hence may be exploited for the purpose of delivering the drugs which need to remain in the window of absorption for a prolonged duration of time. Finally, the nature of the drug will also be acting as an important factor for the designing of the GRDDS. The drugs which impact or with an impact on the gastrointestinal transit time, examples codeine and pharmacokinetic agents like metoclopramide and cisapride increases the gastroretention time. Research on the mucoadhesive gastroretentive drug delivery system. After the focus on how the gastroretentive systems are much more suitable for a particular category of drugs, the literature reports were further recced and it was found out that there is a huge interest of scientists which is going on in the field of development of mucoadhesive based gastroretentive systems. The idea of this literature review was primarily to help us understand that there has been a drastic advancement in the approaches towards the formulation of gastroretentive drug delivery systems. In the era beginning from the 90s or 99s or late 99s, the concept was using of the carbopole-like polymers which were simply made into either the simple systems or single systems or the multiple unit systems for adhering onto the mucosal membrane and delivering the drug for a prolonged period of or prolonged duration of time. Later on, the designing of the system was based on the development of advanced polymeric systems. The polymers like chitosan and its derivatives like thiolated chitosans were coated with polyisobutyl cyanoacrylates and a very advanced system of the PIBCA nanoparticles was designed. Similarly, Higo et al. Designed the tetracycline sucralfate complex which were prepared under acidic conditions and showed excellent mucoadhesive properties and retention on the gastric mucosa compared with the physical mixtures of tetracycline and sucralfate. The attribute of sucralfate to remain adhered to the mucosal membrane was very nicely harnessed by simply making a conjugate or a complex with a very common antibacterial agent tetracycline. This intended to remain there adhered while delivering the antibacterial tetracycline for overcoming any amount of bacterial infection in that particular niche area of the GI tract. Similarly, Schmitz et al. Also highlighted the use of stomach-targeted oral mini tablets for low molecular weight heparin which were prepared by using thiolated polycarbophil, and this thiolated polycarbophil was used as the mucoadhesive carrier material. We also had certain literature reports wherein the extended gastric residence time was obtained by using the positively charged ion exchange resin cholestyramine, an anionic resin due to the adhering to and coating of the gastric mucosa. In conclusion, what we can say is that the research is primarily focused on designing and developing of single or combinatorial approaches for development of various polymer conjugates or complexes in accordance to maintain better mucoretention and to develop systems which will remain adhered for prolonged duration of time in order to release the drug on a sustained manner. Later on, the researches are now focused on development of multiple systems, primarily nanoparticle in nature, which will have certain added advantages and can prove much beneficial in cases where we want the retention and the permeation together in order to get better therapeutic outcomes. My dear students, in summary of what we have just learned in this current module, we can very effectively conclude that the gastrointestinal drug delivery system has got many approaches by which they can be formulated. The approaches could be that of using a floating drug delivery system. We can use an approach of high density. We can go in for the use of many more approaches. Amongst all of them, as per what we have understood, the mucoadhesive gastrointestinal drug delivery systems is the simplest and the easiest formulation approach. We can probably have the GRDDS based on the mucoadhesion, which can be either single unit or the multiple unit GRDDS. The system could be as variant as a mini tablet or a mucoadhesive microspheres. They can be single unit, they can be multiple unit, but all based on a single premise of being mucoadhesive in nature. If we just go back to what we have just learned, is that the GI tract, which is primarily the site for the use of such system or site for the delivery of the GRDDS, is a very dynamic anatomically and physiologically. Therefore, if we are designing a GRRDS, we have to be very sure that we are able to overcome the various barriers which are offered by the GI tract. If we are able to do that and we are able to use the various types of mucoadhesive polymers, we can very effectively formulate systems which can be very successfully used for treating a variety of diseases. The final formulation approach will depend upon the choice of the drug, the disease chosen, and the final therapeutic goal. When we say the final therapeutic goal, it could be either something wherein a drug delivery system is made in a manner to deliver the drug locally, or it can be used in a manner that it delivers systemically. So we have a different therapeutic goal. In conclusion, what we can say that the gastroretentive drug delivery systems, more so the one which is based on the mucoadhesive polymeric base, is a novel approach for site-specific drug delivery, which can be very well exploited for delivering the drug for the best therapeutic outcomes. Thank you.